Just a quick review of the previous lecture so that there is continuity in the lectures and it's easy to understand, okay? We were talking about the complement system, that these are different proteins which are synthesized in the liver. There are about 30 proteins and there may be even more. नहीं आ रही आवाज? Okay, and they are present almost everywhere in your body. They are in the blood, in the lymph, in extracellular fluids, okay? And one of the first weapons to fire by the innate immune system, as I told you, and the most important of these complements, as we discussed in the last lecture, was C3. Deficiency of C3 causes serious problem with pathogen fixation, and a person who is deficient in this particular complement would be prone to develop different bacterial infections because of its very important role here. This C3 in the form of C3b or part of C3b is fixed to the pathogen, and we learned that the C3 complement, it is when it is cleaved into two parts, a bigger part and a smaller part. The bigger parts become fixed to the surface of the pathogen. It becomes covalently attached with the surface of the pathogen, and the smaller part recruits phagocytic cells or other cells of the immune system to come here at this site of infection and play their role. Okay, so the phagocytes would, come in this picture later when we would discuss about the role of the phagocytes, but this was just about the importance of C3. And then I told you that what is the secret of C3 being so important? The secret is hidden inside the structure of this complement, which is actually this linkage, the thioester linkage. Normally, when these proteins are synthesized in the liver, this particular bond or this linkage is hidden in the structure of C3. But when you put C3, for instance, in aqueous environment, in water, then this conformational changes can take place in C3, and water molecules can make bonds with this particular thioester linkage, okay, to make a soluble C3b-like molecule. Or you can say that if you cleave this particular complement, the C3, into two parts, the hidden thioester linkage can come out as a result of that process as well. Either you put it in water or you just cleave it into pieces, and when you cleave the smaller part C3a from the bigger part C3b, this bond is exposed, and this is a nucleophile. It can readily make bonding with molecules like water, or it can make bonding with some amino acids. Both water and amino acids are abundant on the surface of the pathogen, okay? So if it mixes up with water, it can make a soluble C3b. And lots of C3b which is formed there inside your body would be a soluble form of C3b. But some of the C3b becomes attached to the surface of the pathogen because there are amino acids on the surface of the pathogen as well, and they can equally target this particular bonding. So the amino acid would make a bond with C3b and make a covalent linkage. This is also a covalent linkage. This one also is a covalent linkage. And this covalent linkage would fix this molecule, this C3b, the bigger part of the complement 3, to the surface of the pathogen. So this is fixation, and is more and more of these C3b molecules would be fixed to the surface of the pathogen. That would make the pathogen known inside the body to be targeted by different cells of your immune system. In order to destroy it, it is very much needed to coat it with some markers or some molecules. Then I told you about these different pathways: the alternative pathway, the lectin pathway, and classical pathway. These are three different ways which can activate these complement proteins in our body, you know. But I told you that alternative pathway is the first one to be activated. And the goal of all these pathways is to kill the pathogen ultimately by two different processes, either by leveling or coating it so that the immune cells would phagocytose these pathogens effectively, or by just burning holes in the membranes of these pathogens. Lysis of their membranes is another mechanism which can be, which can result as a result of complements activation. So the alternate pathway, I told you about this as well, that C3, where it's synthesized by the liver, inside the liver, it is transported into the blood. The environment inside the blood is a little bit different from environment in the liver. It is more aqueous environment, and in this aqueous environment, conformational changes would take place in this molecule in order to make way for the water molecules to make bonds with C3. And this kind of water bonded with the C3 molecule is called as IC3. Okay? And this IC3 then binds to another factor which is known as factor B. When factor B binds with IC3, it undergoes a conformational change as indicated by this groove here. And this conformational change actually is an invitation for another factor, another complement factor, which is D. And this D would recognize this conformational change and come here and bind to this particular spot and cleave the factor B into two pieces, a smaller piece and a bigger piece. The smaller piece will just go away doing some other job, while the bigger piece will remain stick to this particular IC3. So IC3BB, IC3B is this one, and BB is this one. IC3BB is actually the soluble C3 convertase. But this soluble C3 convertase still is efficient enough to, cleave complement 3, which is abundant there in the blood, in other tissues. So when it comes around with C3 in the surrounding, it's going to cut this C3 into a smaller part and a bigger part, A part and a B part. Okay, so this is a C3. It has come in contact with this soluble convertase here, and it has cleaved it into two parts. Now this is an anaphylatoxins or a chemoattractant, the C3A, and the B part has just bonded to the surface of the pathogen here through covalent linkages. And this part, the C3B, which is now bound to the pathogen, again would undergo the same process, binding factor B, and factor B would be cleaved into two parts, and one part of the factor B would remain bonded to this particular C3B. This is a stable or potent C3 convertase. C3 convertase means an enzyme that can convert C3 into parts, or which can cleave C3 into parts. So in this structure, if you see, this is C3b, which has been fixed to the surface of the pathogen right here. Now it is bound with BB, another factor here. So BB actually is playing the enzymatic function. The later repertoire of events that are going to unfold is because of the BB, or the bigger part of factor B, which is bound here. Now,